All right, protein metabolism. 25-4, I think it is. So protein metabolism. All right, so first of all, let me tell you about this transamination. This is the word right here. Sometimes these transaminases are called, so these transaminations moving an amine group from one molecule to another. These transaminases are also called aminotransferases. You're going to say, when do you use those words? There's no rule. In fact, in the laboratory, for example, in the laboratory, we have this glutamic pyruvic transaminase, which equals alanine aminotransferase. Aminotransferase. So, transferase. So GPT, glutamic pyruvic transaminase, equals ALT. So in the lab, because you can run tests on these. By the way, you run tests on these transaminases because it gives you a snapshot of liver function. A lot of this protein metabolism is occurring in the liver. So this transaminase equals that aminotransferase. Uh, they're just equal to each other. That's all. AST, I forget the other word. I forget the other name for AST. Aspartate. I just forget it. Sorry. Anyway, transaminases equal aminotransferases are the same enzymes. They just have two different names. So what happens is this. If you have an amino acid and there's 20 amino acids, well, there's 20 amino acids that are coded for by your DNA. There's actually, let me talk about that for a second. We always say to you, there's 20 amino acids, there's 20 amino acids. Well, it's not really true. There's really far more than 20 amino acids, but there's 20 amino acids coded for by the DNA and the messenger RNA. There's some other amino acids that are post-transcriptionally modified or actually just exist in some metabolic pathways, like the urea cycle, which we're going to talk about here shortly, has some unique amino acids in it, but they're not coded for by any particular codon. So anyway, you take one of your amino acids, and you're going to take this amine group, and you're going to take it off this amino acid and stick it on another molecule, like this keto acid 1. That's what they're calling this, just keto acid 1. All right. So here's what I do. I take this NH2 and I stick it in place of this carbonyl. It goes through an intermediate called a Schiff base. That's some organic chemistry. I really don't care if you know, but if you do any further research into this, like I know some of you do, you're going to see that this goes through an intermediate called a Schiff base. All right. So this amino group goes over to this carbonyl. Well, what does this amino turn into? What does this position, what replaces the amino? And the answer is an oxygen, a carbonyl. Here's the carbonyl that used to be this right here. It used to be that. And then here's the amino that used to be a carbonyl right there. And what happens is I make a new amino acid. This keto acid 1 became tyrosine, and this glutamic acid became a keto acid 2. So that's what these aminotransferases do. We can take some other organic molecule like keto acid 1, we can stick an amine group on it and make an amino acid, tyrosine. So these transaminases, or also known as aminotransferases, they do this for us. By the way, kind of cool. These transaminases need vitamin B6, pyridoxine. That vitamin is needed for these reactions. All right. We also have deamination reactions where we can deaminate. The reason we typically deaminate is so we can run this organic acid through one of the metabolic pathways like the Krebs cycle or something like that. So what's deamination? Well, all we do is take off that amino group and replace it with a carbonyl, a C double bond O. And what does that amino group become? An ammonium. All right. Now, this is bad. We want to get rid of that. We can't allow ammonia to float around our body. We can't allow too much ammonia to float around our body. It leads to encephalopathy. Encephalopathy. Ammonia hurts your brain. It leads to encephalopathy. So we have to get rid of this ammonia or ammonium. Realize that it's just a protonated form of it. That's all. They're interchangeable depending on the pH of the environment. So ammonia or ammonium leads to encephalopathy. I'm just seeing if I spelled that correctly. It looks all right to me. So we have to get rid of it. We can't let it float around. Well, what do we do with it? We run it through a cycle in the liver called the urea cycle. See, we don't let ammonium float around our body. We convert it to urea. And in fact, 2 ammonias can go into making 1 urea. And then our kidneys get rid of the urea. Now, the urea is no picnic. It's not like we want a bunch of urea floating around our body, but it's better than ammonia. So it's better than ammonia or ammonium, and we run the ammonium through the urea cycle, and we make urea. And then the kidneys get rid of that. All right. So the urea cycle, by the way, the urea cycle is also called the Krebs-Heinzelite cycle, because the same guy that elucidated the Krebs cycle, the citric acid cycle, Krebs, was also involved in elucidating the urea cycle. Same guy. So the citric acid cycle is called the Krebs cycle, and the urea cycle is called the Krebs-Heinzelite cycle. I'm not sure I spelled Heinzelite correct, so I could have spelled this guy wrong, and I'm sorry if I did. We have amination reactions. We have aminases, and that's when you take the ammonia and you stick it on wherever you have a carbonyl. And you can see that alpha-ketoglutarate can be converted to an amino acid, glutamic acid. Now, I don't know if you realize this, because I told you not to memorize intermediates, but alpha-ketoglutarate is an intermediate in the Krebs cycle. You'll find it in the Krebs cycle. So you can take one of the Krebs cycle intermediates and you can aminate it and make a amino acid, glutamic acid, or glutamate. So that's another reaction we have. So we have deamination reactions, we have aminotransferase reactions, and we have amination reactions. With the deamination reactions, we run that ammonia through the urea cycle in the liver to make urea. So this is extremely important. Running excess ammonia through the urea cycle is extremely important because ammonia will hurt your brain. All right. This is the collage showing you all of it. So this is the gist of protein metabolism, moving these amine groups around and ultimately getting rid of the amine groups via the urea cycle, converting ammonia to urea. All right, that's actually the full collage for you, everything that happens. Now, just so you know, you can't make every amino acid. Humans can't make every amino acid through these reactions. So what we have is we have amino acids that we must eat in our diet, and these are called essential amino acids. We can't make them. We must eat them, and I think there's like 9 of them. I'm not a very good nutritionist, meaning I don't memorize recommended daily allowances and stuff like that, and I don't memorize all the essential amino acids. A well-balanced diet will give you all the essential amino acids. If you are a vegan or some other version of vegetarian, you do have to be a little bit careful. You have to eat a variety of different non-meat products to get all of your essential amino acids. And typically, if you're a vegan, then you're probably well read enough to realize this, because all the literature says this. Says, you have to mix your fruits and your vegetables with nuts and things like this. And by the way, mushrooms are a great source of protein, like the portobello mushroom. Mushrooms have high protein, so you can do that. But the typical meaty American diet, you get all your essential amino acids because a steak has all the essential amino acids. So, all right, that's protein metabolism.